everyone. This is group 4, and we will be talking about Salmonella. Salmonella infection is called salmonellosis, and it is a common bacterial disease that affects the intestinal tract. Salmonella bacteria typically live in animal and human intestines and are shed through stool. Humans become infected most frequently through contaminated water or food. Salmonella is most oftenly caused by eating raw or undercooked meat, poultry, and eggs or egg products, or by drinking unpasteurized milk. The common signs and symptoms usually include diarrhea, stomachache, fever, nausea, vomiting, chills, headache, and blood in stool. These signs and symptoms of Salmonella infection generally last a few days to a week. Diarrhea may last up to 10 days, but it may take several months before the bowel returns to usual stool habits. A few variety of Salmonella bacteria results in typhoid fever as well, which is sometimes a deadly disease that is more common in developing countries such as India, Pakistan, and Bangladesh. So Salmonella can be caused by not handling food properly. So many foods become contaminated when prepared by people who don't wash their hands thoroughly after using toilet or changing a diaper or after handling contaminated food, which can lead to salmonellosis. Also, if the surface is infected and someone touches something that is contaminated and then puts their fingers in their mouth, that can also cause salmonella infection. Moreover, animals and pets, especially birds and reptiles, may carry salmonella bacteria on their feathers, fur, or skin, or in their feces. And some pet foods may be contaminated with salmonella that can infect animals. So we need to be careful around animals as well, as they can transmit salmonella to humans. Last but not the least, we know salmonella bacteria live in the intestine of people, animal, and birds. Most people are infected with salmonella by consuming food or water that has been contaminated by feces. So food that are commonly infected by salmonella can be raw meat, poultry, and seafood. So feces may get into raw meat and poultry through butchering process, and also raw or undercooked eggs can be contaminated by salmonella. We may think that an eggshell may be the perfect barrier for contamination. However, some infected chickens produce eggs that contain salmonella before the shell is even formed. Moreover, it is important to pasteurize unpasteurized dairy products because unpasteurized milk and milk products may be contaminated with salmonella, and the pasteurization process kills harmful bacteria, including salmonella. Some fruits and vegetables, including particularly imported varieties, may be irrigated in the field or washed during the processing with water contaminated with salmonella. And contamination can also occur in the kitchen when juices from raw meat or poultry come into contact with uncooked foods such as salads. Prevention can be taken in many ways. First, as always, it is very important to wash hands thoroughly. It is best to wash hands for 20 seconds after using toilet, after changing diapers, after handling raw meat or poultry, after cleaning up pet feces, or even touching pets or other animals and their habitats, especially reptiles or birds. Also, it is important to keep things separate to prevent cross-contamination. Cross-contamination can be prevented by storing raw meat, poultry, and seafood away from other foods in your refrigerator. Maybe having 2 cutting boards in your kitchen, one for raw meat and other for fruits and vegetables. Also, we should never place cooked food on an unwashed plate that had previously held raw meat. Moreover, we should wash food preparation surfaces thoroughly with soap and water.
As mentioned before, the modes of transmission of salmonella is through consuming contaminated food and or water, and even vegetables, like she was saying, if they're washed with contaminated water, or even in the field, if they were introduced to animal manure that contained it, they'll also have some salmonella. It can also be through the human transmission through the fecal oral route. So not washing your hands after using the restroom or changing a diaper and going straight to putting your fingers in your mouth or cooking. And then also contact with animals, which are usually asymptomatic if they have it, so you wouldn't even know. So just make sure you wash your hands after handling animals and pets. So Salmonella is an intracellular bacteria, meaning that it invades a host cell and multiplies within the cell, destroying it. And it has many different serotypes. So that's what the photo on the right shows. Some of the serotypes are host specific. So, for example, the W one is only found in cattle, and that other one is found in pigs. And if you're infected with a host specific one like that, it can be very life threatening for humans. But most of the serotypes have a wide range of hosts and cause gastroenteritis or inflammation of your intestines. And the most common ones in humans are Salmonella enteritis and typhimurium. More severe cases are usually observed in young or elderly people or those with compromised immune systems. Salmonella has an incubation period of 72 hours, but the illness can last from 2 to 7 days. And actually, once you have surpassed the symptoms and you're feeling better, Salmonella can still be found in your stool for months and even up to a year after the infection has run its course. So the way that Salmonella infects your body is it enters through your mouth and it travels all the way through the digestive tract through to the intestines. The first barrier of protection that your body uses are your macrophages, and when the macrophages engulf the Salmonella, they put them into a compartment and try to acidify them to destroy them, but the Salmonella fights back and by increasing the pH or acidifying its own internal pH or cytoplasmic pH so that it won't die and it can actually survive and begin to multiply within that macrophage. Sorry. Salmonella produces both endotoxins and exotoxins. So the endotoxin it has is the lipid A part of its outer membrane, which causes a cascade of biological effects. But it has its exotoxins, which specifically the cytotoxin is a mammalian cell killing type of toxin. And so it will cause the cell to burst and be destroyed, especially once there are enough Salmonella bacteria that have replicated within the cell and they're done with it, then they destroy the cell and move on. Salmonella can find its way through to your bloodstream and your lymphatic system and lead to sepsis, which is very dangerous. And so, Salmonella is actually a worldwide problem, and it's not like a major problem anywhere specifically. It's everywhere. Everyone eats food and everyone drinks water, and not everyone has access to clean water, or not everyone is completely hygienic when preparing food. According to WHO, it's 1 in 4 key global causes of diarrheal diseases, and 550 million people worldwide fall ill each year. And that includes 220 million children under the age of 5 who are very susceptible to severe cases of Salmonella. It's one of the leading causes of morbidity and mortality in developing countries with poor hygiene and a lack of resources to clean and safe water. And there was a study done in Chad, Africa, through certain hospitals in the area. And they were noticing by taking stool samples from the patients with diarrheal diseases that Salmonella strains were present in most cases, but they were not being tested for or isolated as treatment for the patients. And so often the Salmonella was just untreated and antibiotics were rarely given or just not even being used. And then also they were able to find that, especially in young children, the non-typhoidal Salmonella disease is associated with malaria, or can be associated with malaria, anemia, malnutrition, and also HIV infection. And the addition of those diseases to Salmonella can make it very difficult to recover.
To isolate Salmonella, we would first need to transfer 0.1 milliliters of the mixture to 10 milliliters of the RB medium and 1 milliliter mixture to a 10 milliliter TT broth and mix using a vortex mixture. Once the sample has been shaken, we would incubate the RV medium and the TT broth at a temperature-controlled water bath. Once it's done incubating, we would mix the liquid and streak the RV medium and TT broth onto 3 different agars: BS agar, XLD agar, and the HE agar. Keep in mind that the BS plates would have to be prepared a day before streaking and stored in a dark room at room temperature. The colonies will appear differently on each agar. The colonies on the BS agar will appear brown, black, gray, and may have a metallic sheen. We can also see a halo effect on the colonies. Essentially, it is when the surrounding medium appears brown at first, but later turn black when incubated at a longer time. The colonies on the XLD agar will appear pink with or without a black center. They may also appear glossy with black centers or completely black. The colonies on HE agar will appear bluish green to blue with or without black centers. They may also appear large and glossy with black centers or completely black. Keep in mind that the HE and XLD agar plates must be streaked and incubated before examination. There are 3 methods used to diagnose Salmonella: bacterial culture, blood culture, and PCR testing. Bacterial culture tests enteric samples, while blood cultures may be used to isolate Salmonella Dublin or fever and sepsis. PCR methods performs the environmental surveillance test. Enteric samples have an overnight process that assists the organism's growth, recovery, and detection. Moderate to heavy Salmonella infections may be detected from direct plating on the culture media, while light infections need to be incubated. Salmonella can be cultured from a lung or lymph node sample. Bacterial culture has an advantage over PCR testing in that it is able to perform antimicrobial susceptibility and determine the serotype and serogroup. We would first need to run a lab test on a sample of stool, blood, body tissue, or fluids such as blood to diagnose a Salmonella infection. Once Salmonella has been detected, it would be submitted to the state's public health lab for serotyping and DNA fingerprinting. Serotypes are groups within a single species of microorganisms such as bacteria, viruses that share a distinctive surface structure. Salmonella has 2 surface structures that can be separated into different serotypes. The O antigen is the outermost portion of the bacteria's surface, while the H antigen is a slender thread-like structure of the flagella. Serotyping has been used to monitor Salmonella infections for decades and track Salmonella outbreaks. Most people are able to recover from a Salmonella infection without treatment. However, those who have a weak immune system, older in age, or immune compromised may have a severe case and will require antibiotics. When one has been infected, it is important to stay hydrated. Some antibiotics that may be prescribed for this infection are listed.
Last but not the least, here is our physical handout for display. So just to review, Salmonella is a gram-negative facultatively anaerobic rod-shaped bacteria. Sometimes people with Salmonella may experience little to no symptoms, and some might experience an extreme form of diarrhea and other symptoms such as fever, loss of appetite, headache, and nausea and vomiting. Salmonella is something that affects our intestine, and in some cases, it can be extremely deadly if it bursts out of our intestine and is left untreated. Also, a couple of prevention tips, especially if you're working in the kitchen. First, always wash your hands, keep your areas clean, make sure you cook properly, and avoid cross contamination. Common foods that may cause salmonella are poultry, meat, vegetables, or fruits. Surprisingly, salmonellosis annually causes 1.2 million illnesses, 23,000 hospitalization, and 450 deaths in the United States every year. Salmonella is the second leading cause of food poisoning in the US, making an estimated 1.35 million Americans sick annually. So, volunteering at hospice, I saw that salmonella is very common there, and a part of the reason is that elderly are vulnerable because of their immune system are not quite developed enough to protect them from foodborne pathogens or too fragile to offer much protection. Moreover, they don't receive proper care either. I noticed their hands were barely ever washed before providing them food, and sometimes their food wasn't cooked properly or isn't washed properly. So one thing that you all must get from this presentation is that it is very, very important to keep yourselves and your surroundings clean, and in general, clean thoroughly and routinely. So this is our works cited, and this brings us to the end of our presentations. Please feel free to ask us any questions that you have.